Hi everyone, thanks for joining us for the webinar organized by Netri. So we're going to wait a few seconds to have all the participants in the room. And please wait. Welcome to the Netri webinar number 3. We are going to talk today about central nervous system applications and more particularly about inflammatory bowel disease and Alzheimer. So to help me in this webinar, I would like to welcome Benoît Maisonneuve, Jessica Rontard, and Louise Mini. They are all a PhD in either bioengineering or bioscience, neuroscience, and so they will be here to make the presentation. So you have also their pedigree, as we say, their experience and also education. So today we're going to talk about inflammatory bowel disease that will be discussed and presented by Benoît Maisonneuve and Jessica Rontard. Then we will discuss about the Alzheimer disease modeling, again with Benoît, and this time with Louise supporting that. And we will have, of course, a question and answer session at the end. So just to recap a bit the Netri technology, where we're going to discuss about. So you may already have attended some others webinars where basically you have listened and you have heard that our technology is basically organ-on-chip devices that is compatible with lab equipment because it's 96-well plate. You probably have heard also that we have materials that allows not to use pumps, basically, so it's a standalone device. And finally, the chip that we are, and the design of a chip helps to have compartmentalization of the cultures and also some very smart way to link them together through axon projections. So we can go to that in a few seconds. Additionally to that, you also probably know, or you will know, that we have a unique proposition in terms of biodigitalizing the human body using the neurons as sensors, and that would definitely give you a wide scope of applications. You're basically addressing different organs of the body and then the therapeutics related to that. Then, of course, you probably know already that we use a specific technology that are dynamic but also non-destructive, like, for example, the electrophysiology. And you would see here on the right, on the digital library part, that basically we are capable using reference compounds and testing in yellow here, compounds to test. We are capable to find some predictivity in terms of efficacy and toxicity. So that's also unique. So as a summary, the technology is basically made to replicate the human body and the natural compartmentalization that is linked with. Like, for example, here on the scheme, you have sensory neurons that comes from the finger, where you can touch your skin, and then you will get information into your sensory neurons that will go up to link to the nervous central system then. So basically what we are really doing here in our chip is two things. The first one is being able to culture separately organs in different channels, in 3 channels, if you'll follow my mouse, 1 channel here, a second channel here in the middle, and a third channel here that are separated. They are physically isolated through the second channel. Additionally to that, they have an inlet and outlet well. So each channel can have pipette in and pipette out. So that helps also to collect information like supernatant and do analysis based on that. So where is the beauty of our technology is, as I said here, is the channels here do not see each other's. They are 3 channels separated. They can do co-culture 3 culture depending on the size of the channels. What we have invented and developed and then produced now commercially available in 2D architectures is the ability to make micro channels that are extremely small, about 3 by 5 microns section, that allows only the projection of neurons that to follow those guides, those micro channels. So if you take basically cells body here of sensory neurons, and you see the projection and the neurites, they will possibly use the microchannels as guided and will cross from the channel 3 here on the right to the channel 2. And then they will take again microchannels that will help them to move from channel 2 to channel 1. So that will give us actually the ability to measure the difference between relatively measure reference compound versus a compound to test. And so here you have, for example, from our last webinar, we talked about the traumatic nerve injury and basically doing axotomy in the middle and see how, based on different molecules and concentration of molecules, how the neurites can grow again. So that's something that you will be able to see as a replay from our last March 19 webinar. The other thing we talked last time was also the innervated skin and the proof of concept, basically using our technology of co-culture. And here, for example, having sensory neuron on the left here and crossing over keratinocytes and building a 2D innervated skin model proof of concept. You have also some indication here that we can basically, in one chip, we can have several informations. The first one is the vehicle can be used before to make sure that it works correctly. Then the compounds will be tested. And at the end, we will apply some positive control to see that there is no damage, that the chip is reacting correctly after the compound is testing. So again, this information is available on our replay on our YouTube channel in the March 19 webinar. So having said that, as a recap, now we can start to move to discuss about inflammatory bowel disease. And I will leave the word to Benoit. And Benoit, up to you.
Thank you, Thierry. Thank you, everybody, for joining us today. So as Thierry mentioned, today there will be 2 main topics. The work we did with various partners, one which is the CHU of Saint-Etienne in IBD, and the other one with ETAP-Lab on Alzheimer disease. So jumping straight away into IBD. So just a quick reminder for those of you that are not familiar with IBD. So IBD are very complex chronic inflammation conditions that affect the digestive system with 2 main distinctive entities. So there's the ulcerative colitis and the Crohn's disease. They are characterized by very intricate inflammation of microbial dysbiosis, sorry, that's a complicated word for me, with neurocognitive symptoms. It's a very interesting disease for us to study because it puts into play various organs and various parts of the body that our technology enables us to recapitulate and study. One of the main components of those diseases are the neuroimmune communication with the release of pro-inflammatory factors and also the modification of cell-to-cell interaction. There's a lot of challenges in studying those diseases, mainly because they are, as I said, highly complex, because they do require complex organ modeling. And that's where actually organ-on-chip can make a big difference, because they can enable researchers and pharmaceutical developers to actually have better tools to ask questions and get answers that they could not get before. So here in the case of IBD, of course, the gut-brain axis is very central to the whole thing. So when we think about gut, well, obviously we do have the gut, but the brain part is extremely important. The whole nervous system is very central to how the overall thing behave. And so actually in order to start modeling some aspects of those diseases, we choose to use our dual link shift MEA. So that's one of the devices that Cherie showed you before with 3 compartments. And that enabled us to actually have a culture on either side, so in channel 1 and channel 3, so the channel that is on the left side and on the right side, and have a synaptic compartment in the middle. That's what it looks like, basically, the device by themselves. So we have 2 different ones according to the equipment you may have in your labs, but that's basically what they look like and really match 96 well plates. So of course, the first thing we do when we work on this kind of modeling, so once we decided of the device that we have that will be the most adequate, is to start to define the neurons the cells we're gonna need. Here we use glutamatergic neurons from brain cells. So here you can see I'm not going to go into depth in this one, but you can see some data of the overall adaptation of that we did from the supplier culture protocol in order to get good culture in our devices. So the good thing is we have very similar results to what we'd be expecting when we look at wells in terms of viability and expression. And that enables us, when coupled with the MEA, to actually look at the electrophysiological activity of the neurons. So if we dig a little bit deeper, so as I mentioned, the work I'm going to talk about very quickly today has been done in collaboration with the CHU de Saint-Etienne. And you have the reference here, it has been published last year. And it really focused on neural communication between glutamatergic neuron and immune cells and dendritic cells, to be a bit more specific. So I'm not going to go too much in details in what we did and how we did it, because you can actually have all the information very well detailed in the paper. But basically what we did is we had a co-culture. So we had a culture on the left side, glutamatergic neuron, same thing on the right side in channel 3. And we added some dendritic cells either in channel 1 or in channel 2 or in channel 3. And we also tried to stimulate the glutamatergic cells with LPS. So here you have an example of the cell culture. So on the left-hand side, the 3 images you have show the human-induced pluripotent stem cell-derived glutamatergic neurons in the devices, and with regular markers that are expected from them, like MAP2, etc., just to make sure we see them. The interesting part is on the right-hand side, when we decided to add dendritic cells in the channel 1. So if you look at the immunostain here, that's the little green dots you can see in the channel 1 on the left-hand side of the image. And just below that, you have images just of what the culture looks like before the dendritic cell seeding and after on the right-hand side, when you can see that they're a bit everywhere. What we did afterwards was, well, a few different things. So if we go step by step, if you look at the top left corner, the graph at the top left corner, what we did was an ELISA in order to measure the quantity of IL-6 that was produced in the device under various conditions. So we checked that the neuron by themselves do not produce any. When stimulated with LPS, they do not produce some either. However, when we co-culture them with the dendritic cells, we could detect very high level of IL-6, which is good, which was expected. The very interesting thing as well is what you can see here, for example, is if you put dendritic cells in the channel 1, you don't see expression or detection of IL-6 in the other channel, where that showed that the compartmentalization works really well to prevent the diffusion of compounds from one side to another. On the right-hand side, the graph on the right-hand side, what you will be able to see is the VGLUT1 quantification from the immunostaining, and what you can see that both LPS and addition of dendritic cells increase the expression compared to baseline. And at the bottom, you have 4 graphs that represents the functionality of the glutamatergic neuron with or without the dendritic cells. So that was recording using MEA. So the functional activity really that can be expressed here was expressed using the mean firing rate. So that just corresponds to the number of spikes per second during the recording. And what we show was that the change in MFR compared to the baseline, when we add dendritic cells to the glutamatergic neurons, we had an increase when we added LPS, and we actually could see decrease when we were adding some dendritic cells, which again was to be expected and validate the mechanics that we were expecting to see within the model. So that's very interesting because it shows that there is a pattern of electrical activity in the presence of the neuroimmune cell co-culture, and that indicates there is an increase in nerve impulse when the dendritic cells are present in the channel 1 with the neurons. Just to conclude very quickly on that, so this proof of concept, basically, study that allowed us to show that we can create a multimodal way to assess immune and nervous communication, so using electrophysiology, supernatant, and everything. What we have shown as well is that the transduction of inflammatory immune cell signals to neurons leads to an electrical impulse materialized by an increase in firing rate in neuronal culture. So this signal is bottom up, and that's consistent with the intuitive neuron communication within the intestinal mucosa. So from that, I guess one of the questions you may have is what are the next steps? Because there are some next steps. And that's actually complexifying the model by adding some intestinal components. So that's work that is ongoing at the moment when there are several axes that we are exploring, such as adding an endothelium layer in order to better model the intestinal work, the intestinal parts. And also then we can explore a lot of various other conditions like with using specific bacteria and etc. Moving on to the Alzheimer disease. So the Alzheimer disease model, the work I'm going to present to you was realized as part of a project that was financed and that is currently ongoing called, under the big financing called France 2030. And so that's the project we call Biodiamond, and that project was and is realized with a partner called ETAP-Lab, which is a CRO that is specialized in vivo work and in developing peptides in order to create disease models. So the way we approach this kind of model, so we just a quick summary. I guess I don't really need to go into much detail because I'm fairly sure that most of you have an idea of what Alzheimer's disease is, but it's one of the most common degenerative diseases that leads to declining memory, cognition, and various brain functions. There is a growing body of evidence that suggests that the oligomeric form of amyloid beta and tau are key players in this pathology. At the moment, when you look at clinical development, there is very high failure rates and very long development time. That's all due to several factors, such as a very poorly understood etiology of the disease, but also because of physiological differences between the species when researchers try to understand what's happening in the brain. So that's where actually brain-on-chip platforms can be very, very efficient, because that enables us to use human cells, and so remove the problem of the translation of the preclinical model, and also to ask a very specific question, because it allows us to study and image and look really in depth into the mechanism. So just very quickly, and once again, I guess you're all aware, well, the brain is a very complex organ, and it is made of several regions. And that's one of the reason why we believe that our technology is very well adapted to study this kind of organs, because it is, because of the compartmentalization, inherently able to have different population, and you can control the way they communicate between themselves. So, of course, as before, we choose to work with the DualLink, our DualLink MEA. So we had two that we are using mainly, which are the regular MEA and the Shift MEA. And once again, that's what they look like. I guess you are aware of this. Same thing that before. The first thing we do is to actually adapt protocol from cell suppliers when we find cells of good quality. So here it's very similar to the one you've seen before. The only difference is that on one side you have glutamatergic neuron, and the other side you have GABAergic neuron. We can culture them. We can see the expression of what we're expecting to see at various time points, and we can see activity happening throughout the culture until it reaches a maximum, and they are ready to be used from day 21. So going a little bit more in the culture model, so that because of the type of cells we decided to use, this is in a certain way a healthy one, meaning that there is no component that is linked to the disease at this stage. Because first we need to make sure that we have a robust culture, and then we can move on to the pathology part, and I will get back to this a bit later. So as I said before, we decided to use the TriLink, and we have a population. So on one hand, in channel 1, we have some glutamatergic neurons, and in the channel 3, the one that is completely on your right-hand side, we have GABAergic neurons. Here you have an example of what it looks like when in culture. So with, once again, the glutamatergic on the left-hand side and the GABAergic neuron on the right-hand side. When we looked at those immunostaining and we start to quantify various markers, as expected, we can see very low markers such as SOX2 and nestin, which show of immature actually stem cells, and we can see high level of VGlut1 in the glutamatergic neurons and high level of GABA in the GABAergic neurons. So all of this was as expected, but that's important to verify it when developing or transferring a new culture onto a new device. As I mentioned before, we also recorded the mean firing rate that is occurring spontaneously within the culture at different time points, and you can see that we start to detect some signal from day 7 and it gets bigger at day 14, and from day 21, that's where we consider we can actually start to explore. As an example, just below the 2 graphs, you have 3 raster plots. So basically, raster plots, they are little dots or spikes, activity basically, and the colors you can see are actually the various channels. So on the top one, you will have channel 3. On the bottom one, the blue bottom, at the bottom is channel 1. The 2 in color you have in all the micro channels. So Thierry mentioned them before. They are the very, very small channels where only the axons and the can go through. And there's the one between channel 1 and channel 2, which is in the middle, and the one between channel 2 and channel 3. So that's good because that showed that we had a robust on-chip cell differentiation protocol, and so we were ready to move on to the next stage, which is to start to work on the entry model. And that's really where our partner ETAP-Lab comes in, because he is quite an expert in developing this kind of peptides. So the work I'm going to present here has been carried out at ETAP-Lab. And so what they did first, they have beta oligomer, of course, and they started first because we were developing adapting the cultures in-house here at Netri. They started to test them on rodent neurons first. The way they did it, they put the neurons in the channel 3, and then they let them grow all the way through channel 1. What here you can see at the top immunostaining of those cells. So you can see the regular one basically on the top left-hand corner, and in the right-hand corner you can see the one when they added 15 micromolar of the peptide, and you can see very significant axonal generation happening. What is very interesting when you quantify it a bit more properly is that when you add that we see axonal degeneration in the channel 3, which is as expected, and we can see it actually reacting in a dose-dependent manner, and that's very important for the mechanistic side. On the other hand, there was no significant death in population in the neuron, and that's good because if you start to have death occurring in the other channel, it means that you're going to bias your study. So it's important to find the right dosage in order to make sure you have the degeneration without causing the cell death right away. So having all those things done, well, the next step, of course, is to bring it together first. So we've developed the culture of human cells, they've developed the disease model, and now we are working currently in bringing the two together in order to have a human-based disease model. So that's something that is happening at the moment, and hopefully you will be hearing about it very soon. Of course, some of the other steps that we have, because the project BioDiamond is wider than just Alzheimer's disease, is to work on other pathologies, and the work is ongoing. So there's Parkinson and ALS. So like same thing, we are developing the cultures in-house and also with other partners, which are cell providers. And our partner ETAP-Lab developed the various peptides that enables us to develop the specific disease models. I think that's pretty much it for me. Thank you.
Thank you, Benoit. Thank you very much for the talk. So we have time for questions, and there are some questions popping up. The first one is about to describe a little more the chip that you have used for IBD. Maybe you should come back to the to one of the slides. Yes. Maybe you could.
I can.
Yeah, please.
Yes, perfect. As Benoit mentioned before, we used an asymmetric devices. So it's composed of 3 compartments called channels in these pictures. So in channel 1 and 3, we had human glutamatergic neurons and separated with macro channels to allow the 3D isolation and also the isolation of soma and axons. The channel 2 is asymmetric channels. So the distance between the channel 1 and the channel 2, it's around 100 micrometers. And the dendrite and axons can pass 4. And the distance between the channel 2 and the channel 3, it's around 500 micrometers. So only the axon can pass so the channel 2, it's modeling the synaptic compartment. I mean, only axon from the channel 3 and axon and dendrite of the channel 1.
Thank you, Jessica, for commenting that. Then we're taking questions as they come, so feel free to add new ones whenever you want. So there is a question regarding Alzheimer part. From the images, it looked like the GABAergic neurons have a worse adhesion to the substrate. Do you think you need to use different coatings for different neurons? First question. And would your chip allow to use different coatings for the different chambers? So maybe we can comment on that.
Just, we can go back on the slide so we have them in front of us. Yes.
This one, I think.
This one would be better. Do you want to start for the adhesion part?
Yeah, I can. So GABAergic neurons in that picture have had a density, a lower density than glutamatergic neurons, and it's physiologic in these neurons. We can use different coating from different neurons because we have the fluidic isolation between the channel 1 and the channel 3. So we can totally use different coating, different media for the neurons. In this culture, we use the same coating because we used a different substrate because we have microelectrodes array in direct contact of the neurons. So in fact, because of the substrate, we need to use a particular coating. I think
Thank you, Louise. And another question is about, can you identify axon from general neurites in vitro without immunostaining? So who wants to answer the question from the team?
I can answer
Go
the
ahead,
question. It's a little bit difficult. You can add it if you want. It's difficult to identify neurites from axons. I think you mean dendrites from axons. Dendrites are shorter than axons, so we can estimate the more axon is longer, there are axons, but it's difficult without staining to identify precisely if it's axon or dendrite.
If I may add on that, so that's also one of the reasons why we also have the asymmetric device with the synaptic compartment in the middle that is a different distance from channel 1 and from the channel 3. The idea is if you create enough distance between two channels, only the axons will be able to cross over. Whereas with the other one, where it's closer, well, they can both go through. So you have the synaptic chamber that is created that way. But yeah, it's difficult to see without immunostaining just from your eye while they're growing.
Thanks, Benoit, and thanks, Louise, for responding to that. Some other questions coming. Are you able to perform automatic measurement the firing synchronicity of the network? Who wants to answer that?
Yeah, I think I can answer. If I understand well the question, you want to know if we can measure the synchrony of network. So we used with Axiom Biosystem device. So we used the software of Axiom Biosystem, and the software can generate several metrics, and we measure the spontaneous activity of the neurons. So yeah, we can see and we can measure precisely the synchronicity of network and of channel 1, channel 3 of the both channel.
Thanks, Louise. Another question, probably around that, but not exactly the same. Can you identify the amyloid propagation / seeding through your devices from channel 1 to channel 3?
Yes, it's a very good question. Actually, we don't perform this, but of course it's possible using immunostaining or something like that.
Or ELISA.
Yeah.
So yes, it's possible. We can identify that. There are some questions about if you have tried adding astrocytes in the co-culture. So maybe Louise, you want to answer that?
We didn't try to add astrocytes, but as Benoit said earlier, ETAP-Lab is working on the subject either, and they tried to put astrocytes with glutamatergic neurons, and it shows that it's improved the electrophysiological activity, but it's well known that astrocytes improve the activity of the neurons.
On top of that, if I may add, so that's something we definitely consider, because we do believe that having astrocytes is extremely important in order to model this disease properly. We're also working with some cell suppliers at the moment where they are developing culture protocol for their astrocytes that's actually all patients or mutated to have the right mutations in order to move forward on that subject. That's a really good question, thank you.
Okay, there is another question about the Alzheimer disease. So is there a PDMS absorption of the oligomers in your device?
Yeah, it's really well known that PDMS has absorption. Everyone knows that. So to avoid the absorption or to control absorption, we measure and we had some tests with a different concentration of the peptide, and we used the best concentration to have the perfect impact of the neurons.
What we could see when we worked with our partner on this subject was that we just had a slight shift in concentration in terms of effect if you look at the dose response curve. But once you perform the shift on one concentration, you can actually predict the way it will behave for the rest. So yes, it's happening, but there are ways around that makes it not an issue at all actually to develop the models.
Thanks, guys, for the answers. We stay on the Alzheimer disease, and it says the model for Alzheimer disease is in development, which is going to be the next one, and the question are Parkinson, question mark, ALS, question mark.
Yeah, in the project of the Biodiamond projects, so we focused on Alzheimer, Parkinson, and ELS. So the next step is ELS with a co-culture of motor neurons and muscle cells, and we want to use a peptide involved in the disease is TDP-43. So we want to do the same experiment than Alzheimer, but on the ALS disease.
Thanks, Louise. Question on the beginning of the presentation about the IBD. The culture medium used for maintaining neurons is different from the medium for maintaining dendritic cells cultures, isn't it? And also, did the dendritic cells survive during the co-culture?
So we use the same cell culture media. It was a neuronal cell culture for both culture. And the second part, sorry?
And the second part was, did the dendritic cell survive during the co-culture?
The aim of this project was to establish an approach to study the communication between neurons and dendritic cells. So we only study the neurons' viability and functionality.
Yeah. Okay. Excellent.
How long did you keep them in touch together?
Only 4 hours.
Yeah. Okay.
Yes.
So it's a bit difficult to answer the question.
Yes, exactly.
Viability.
Okay. Regarding IBD, there was a short question. How was the IL-6 level measured?
Yes. As Benoit mentioned, we analyze the IL-6 level directly in the supernatant using ELISA. And what we quantify, it's an increase of IL-6 only when the glutamatergic neurons are co-cultured with dendritic cells, and no increase with LPS addition or only with human glutamatergic neurons.
Okay, thanks very much. Still, we have one question here that's popping up. Still IBD, why was LPS used in the experiments? Maybe you can detail that, Jess.
So LPS is a lipopolysaccharide. It's a major component of bacteria membrane. Yeah. Sorry. And we used LPS to modeling the inflammation present in the mucosa of EBD patients. So concretely, we exposed dendritic cells 12 hours before sitting on the chip with LPS.
Thank you, Jess, for that. So we're almost there. So if there is questions to pop up, feel free. We're almost reaching the end. So I'll give you a few seconds more. Of course, there will be some more questions that you can ask afterwards. And one of the questions was also, what are the limitations of the Alzheimer disease model? Who wants to answer that?
I can start.
Yeah.
Okay, you go.
So in our model, the principal limitation of the AD model is we used only one cell type. So we used only glutamatergic neurons. Or we know that Alzheimer is involving with several neurons like gluta, GABA, astrocyte, microglia. So to have a more complex model, we have to use another cell types and have a more physiological model, pathophysiological.
Yeah, if I may add a little bit, so definitely, as Louis said, so we have the gluta-GABA co-culture. That is the first step, but it's definitely not enough to model a disease that complex. Another limitation that can occur is, as I mentioned, the way we're moving forward within this project at the moment with our partner is to use their peptides. For example, the A-beta injury. And so that, of course, means that if for whatever reason the researchers in 5, 10, 15 years realize that actually, for whatever reason, even though it's very unlikely, A-beta is not actually a really good candidate, well, it means that we didn't focus on the right thing. So I focused a lot on amyloid beta here. I just mentioned it very quickly, but of course we have other injury models that we're exploring at the same time, like the tauopathies and things like that. So there is always a limitation linked to that. I guess every time you're going to create a model, there will be limitations because they are by definition models. And I think asking the questions like you did, like about the limitations, is a very good way to approach them and make sure that they are the right tools to answer the questions that you have. So thank you very much for this question.
Thank you for all those good and nice questions. We're almost there, so if there is something popping up, you may want to ask it directly in live while we have our team of experts here. Otherwise, it is possible to send questions afterwards. To let you know that there would be a replay of this webinar for all the persons subscribing. And basically, I would like to give you in advance our next exciting webinars topics that we will come in middle of May, the 14th of May. We will introduce the 3D architecture for talking with Walid Duplexwell, but we will discuss about cerebral organoids growth and irrigated skin. That will be a topic that will rely also on some publication and posters that we have done in the past. And then we will have the same month, it will be a loaded month in May. We will have at the end of May some additional information that follows the first webinar we did in February. We talked about pain and pain models, and now we will talk to you about additional features. Pain on MEA, which would be a breakthrough in our current drug discovery applications. So that would be the 30th of May. And then possibly, if not before September, but in September, we will have introduce the digital library, which is coming from our, smart way to measure through electrophysiology and how to be able to predict. I was almost going to leave, but there are a few questions. One question we can take. Do you have extra data available on what's happening in channel 3 after amyloid treatment in channel 1 and publication of poster? We will basically. I see that my moderator is telling that she would respond to that in the chat. So we will do that to the person that asked the question, and we'll send the information. So that's not a problem.
And as a spoiler, it will be basically in the form of a poster at the moment.
All right. I think we should be there. Again, more questions, don't hesitate after the webinar. Thanks very much for attending the meeting. We look forward to see you in May 14th for talking about a breakthrough in 3D architecture or new models talking about cerebral organoids and irrigated skin. Thank you so much for sharing this with us.
Thank you very much.
Thank you, bye.
Have a great one. Bye-bye.